Thank you very much for the introduction. So before I get into talking about grizzly bears or the biomedical promise of hibernation, I want to start by thanking everybody that has worked on this project with me, because working with such large animals is really hard, so it truly is a collaborative project. I'd like to highlight my PI, if I can get this to work. Anyways, right there, Joanna Kelly, who I just found out this morning through tweet. She is looking for a postdoc to work with grizzly bears, so if you know of anybody that's looking for a postdoc, please send them her way. And then I'd also like to highlight Liz down here with PacBio, because she really helped me out with processing this data for this talk. And I'd like to thank the University of Delaware Sequencing Corps and PacBio for selecting this for the 2018 Smart Sequencing Grant. And also for inviting me out here, not just because this allows me to delay my prelims another 2 months, but because I like talking about grizzly bears. All right, so let's get into it. So as we all know, or I hope we all know, brown bears or grizzly bears undergo an annual activity cycle. So what this looks like is grizzly bears, will just start in January here, are going to be right in the middle of their hibernation season. Right around April, they're going to be coming out of hibernation, moving into this active phase. And then right around August, September, they're going into this thing called hyperphagia. And then October into November, they're going back into hibernation. So I just want to slow down here a little bit and talk about the physiological processes or changes that are going on during these seasons. So during hibernation, grizzly bears reduce their basal metabolic rate quite a bit, up to 75%. Coupled with that, they have slower heart rate and respiratory rate, and they are fasting during this time. So they're burning fat as their main source of energy. And then also during this time, any females that are pregnant are going to give birth, and those cubs will stay with them in the den until they emerge sometime around April. Now, during active season, this is what we think of as just normal bear physiology, right? They're just out doing bear things. So this is when they're packing back on lean mass that they lost during hibernation, and they're also mating during this time period. Hyperphagia, right around August, is when they're moving into this. This is really preparing for that hibernation. So they're consuming just an incredible amount of calories, trying to pack on that fat that they're going to need to survive yet another cycle of hibernation. Actually, one of my lab mates works out in Katmai National Park in Alaska during this time period, and he has actually witnessed a bear eating over a 7-hour period over 100,000 calories in salmon. So they are constantly eating during this time. It's actually quite impressive. And so what does this actually look like for the bear? So here is a captive bear. Her name is Luna, and this is her coming out of hibernation, so right in that April time zone. And you can see she's not really that fat. She's quite thin for a grizzly bear, and that's because she's been burning fat the entire hibernation season. This is Luna going into hibernation in October. So you can see there's just this drastic change in body size where they become basically morbidly obese right before hibernation, and they do this every single year without any negative consequences that if a human were to do this, we would expect some pretty drastic medical issues. So I want to get into why we're studying grizzly bears for this as an ideal model for human diseases, because while the physiology is fascinating, I really like the applied side of this. So that reduced metabolic rate up to 75%, the reduced cardiac output, and they drop their body temperature by between 4 and 7 degrees is really great for patients that had a traumatic injury and need to get to the hospital, right? It'll extend that critical window where that patient can seek medical care. Because their metabolic rate will be reduced, they can take more time to get there. They also don't urinate or defecate during hibernation. In fact, their kidneys are basically shut down during hibernation, and yet they don't suffer from any of the negative side effects of that if a human's kidney were to shut down, you would see, like uremia, where they have high urea content in their blood and they eventually die. They don't have to deal with that. The two that I'm really interested in personally are the lack of muscle atrophy that we see in grizzly bears. So although they're laying around for 6 to 7 months out of the year, really not moving all that much, their muscles just don't atrophy at the same rate a human would. In fact, if a human were to lay around the same amount as a grizzly bear, it's roughly equal to bed rest, and we would expect to see about 90% strength loss in a human over that same time period. For a grizzly bear, it's about 20% of their strength is lost. And then during hibernation, grizzly bears are actually insulin resistant. They mimic basically a type 2 diabetic patient during that time period, and then come active season, they switch back into being insulin sensitive and are able to respond to insulin releases from the pancreas. And we'll get into how we know that in just a little bit. So I want to just highlight that bottom point a little bit more to talk about, the human impact of this. So like I said, they mimic type 2 diabetics while they are hibernating because they're insulin insensitive, and that's a disease that's really expected to keep rising, as we all know, as obesity continues to be a problem in our society. On the slide here, I have the world stats, but I just want to bring it a little closer to home with some US statistics. So diabetes is actually the 7th leading cause of death in the United States. It kills over 75,000 people a year. And so 29 million Americans are estimated to suffer from diabetes, and that's going to continue to rise. And this ends up costing the US economy around $245 to $250 billion a year, both in lost wages and in medical expenses. So it's really important that we start looking into this because really the treatment options are limited. They haven't been improved since like the 70s when we started injecting patients with insulin. And so really the only option these patients have are just keep injecting more and more insulin so their body eventually responds. So we really need to find these new approaches, and hopefully by studying grizzly bears, we'll open up these new avenues of research into human treatment. Now, like I said before, studying grizzly bears is really difficult, right? They're very large animals, and they're relatively long-lived. They live about 30 years. So really you have 3 options if you want to start sequencing any tissue from grizzly bears. You can capture them in the wild, but if you want to pair that with a hibernation study, that means you have to capture them both during hibernation when they're in a den, pretty hard to find, and also in active season, recapture the same bear so that you can compare those tissues. Not great. There's a lab in Finland, I think, that does that. We could work with zoos, but zoos understand that these animals are very charismatic and that people come to the zoo to actually see these bears, so they don't allow them to hibernate. They'll feed them through the hibernation season and not give them bedding, and they'll keep the bears up and active that entire time period. So not great if you want to do a hibernation study. The last option, and probably the hardest to convince anybody to do, is to build a bear center to keep these grizzly bears captive so that we can sample them throughout the year whenever we want. Luckily, I didn't have to convince WSU to do that. One of my collaborators did that a couple decades ago. So at WSU, we have this really cool resource, which is the WSU Bear Research, Education, and Conservation Center, where we have 11 captive grizzly bears, and we give them hundreds of acres to roam around so that we can hopefully give them enough activity that it mimics their natural environment. And so we are able to just sequence these bears whenever we really want, which is really a great resource. Now, what's really impressive about these bears is they're actually trained quite well. And so what does that actually look like? So here's a bear that's giving a blood sample. They climb into this crate just for our protection. They're fed a sugar treat that they absolutely love. They actually fight to get into this crate because of how good that treat is. And then we actually tell them left side, right side, they'll lay down, and then they'll present their paw outside of the crate where a vet is taking a blood sample. And this is actually how we show that bears are insulin resistant during hibernation. So here's a graph. We have a dextrose feeding, which is just a type of sugar. We feed them at time 0 on the x-axis. We have every 15 minutes a blood sample was taken, and then on the y we have the blood glucose. And you can see the blue and green lines are during active and hyperphagia. And so there's that spike that we expect when you feed sugar, and then that decrease in blood glucose level as that insulin is released. For hibernation, there is really no quick spike in blood sugar. It just continues to climb, and then eventually it starts to go back down, but not as quickly as we would expect from an insulin-sensitive animal. In fact, it's most likely through insulin-independent mechanisms that the sugar is entering the cells. So how am I using these trained bears for my research? So for this study, we had 3 bears, and we sampled them. We sampled them in January, right in the middle of hibernation, and May, right when they're in active season. And we sampled 3 different tissues. We sampled fat, liver, and muscle. For this talk, I'm just going to focus on liver because that's what I have data for currently. And we sent these tissues out to actually the sequencing core here in Delaware. So thank you very much, Bruce, for working with very small samples because we used a needle biopsy, which if you haven't seen one of those before, it's about the same amount of tissue as you would like if you have a click-top pen. That little clicky part at the top of your pen, that's about how much tissue we sent to Bruce. So he was able to get us really cool results. We used the Iso-Seq method. We got these really long length reads where we're trying to capture the transcriptome. We mapped those reads back to a reference genome using some splice-aware, any of the splice-aware software packages that exist. We filtered and identified new genes and isoforms with the Squanti2 package. And then we are currently working to annotate the function of these new isoforms. So what did we get out of this? So let's do a quick transcriptome comparison. So here's the reference genome that existed prior to PacBio. We have a total length 2.3 gigabases, contig N50 of 314 kilobases. So not terribly contiguous, but a decent genome for a novel model. And importantly, they had 43,168 proteins identified. Now, after PacBio, we didn't improve the contig N50 or anything like that, but we identified 10,633 unique genes just in the liver. So these are genes that are being actively transcribed in the liver. 679 of those genes are actually novel. And so I haven't done much looking into these genes, but I do want to highlight the fact that most of these novel genes are actually just one isoform. So here on the X, we have the annotated and the novel genes, and then we have the % on the Y, and you can see that orange is just one isoform, makes up the vast majority of our novel genes. Really not that interesting to me at this time point, so I want to move on. Unique isoforms, we actually identified 46,248. So what's really surprising about this is just liver, right? So we have more unique isoforms identified than in the reference genome. Right, so we've already expanded that reference transcriptome quite a bit just using the liver tissue, and I'm excited to see what we get out of the muscle and the fat moving forward. Now, what does that actually look like from a quality standpoint? So blue, we have the reference, and in orange, we have PacBio. And so you can see our length of our transcript is actually more realistic to what is biologically known, right? We have right around 2,500 bases is the length, whereas our reference prior to PacBio was absolutely terrible with that spike at the beginning and really jagged moving on. These novel isoforms can then be quantified using Squanti. So Squanti classifies these in a few different bins. I'm just going to highlight 4 of them here. So we have full splice match, which is pretty self-explanatory, right? We have the reference up here. The full splice match matches the reference perfectly. We have incomplete splice matches where we're just missing the first exon right there. The novel in catalog, so we are now getting into these really cool novel isoforms that didn't exist previously where we splice out one of the exons. And then the novel not in catalog, where we're using a new splice site to get a new isoform. So how many of these did we get? For the full splice matches, we have 13,470, and for the incomplete splice matches, we have 8,605. This is a really great check to see if this is working appropriately. It's really nice to see that about half of our transcripts are already identified in the reference genome. Now, the novel in catalog, so the ones that just have an exon spliced out, we have 14,596 of those. And the novel not in catalog, 8,776 of those. These are really exciting because this is where we're going to start seeing all of this new stuff that wouldn't have been captured in the original reference. Now, if you're wondering, well, how many of those are actually biologically relevant? Squanti will output, how much is coding. So on the X, we have the different categories. On the Y, we have % transcripts. And then the shading, the darker shade is coding, and the light is non-coding. So you can see that actually quite a bit of our isoforms are coding, meaning any analysis that was done prior with the reference transcriptome, you're missing a lot of information that PacBio can now provide. And so this was great to see. It was also a little concerning because I had just published a paper 2 weeks ago where I did a differential expression analysis using just the old reference, and I wanted to make sure that it wasn't garbage. So I remapped the short reads that I got for that analysis. We just looked at the liver. We had the RNA short reads from that previous analysis. We mapped to the new reference genome using that PacBio improvement, using Callisto, and then we quantified the transcript abundance using Sleuth, and then we're still working to annotate the functions. So what did we get? So first off, really promising, 38,392 transcripts passed our filter for lowly expressed transcripts. So they had to be expressed at a certain level in order to actually be included in this analysis, and that's how many we included. Out of that, 2,200 roughly transcripts are differentially expressed between hibernation and active season. That's really cool, and a lot of the ones that are differentially expressed fall out also in the previous analysis that I did, that I just used gene level. So that's really nice to see that the paper I had just published isn't garbage. 86 of these genes have isoforms differentially expressed in opposite directions. Now, this was really cool for me to see. This is something that really we did not pick up on because we just used a gene level analysis on the previous paper. And now we're able to see that 86 of these genes, one isoform is up, one isoform is down, meaning possibly that these isoforms are performing different functions between the seasons. And so I want to walk you through one of these genes just to show you, what it looks like. So I chose Cystathionine-Beta-Synthase for a few reasons. It highlights what we can do with PacBio. It also highlights some of the challenges that we face using PacBio. I chose this one also because it has multiple isoforms annotated in other species. However, in the grizzly bear prior to PacBio, there was only one isoform. So it was something that we missed in the original analysis using just this one isoform. In fact, that one isoform that was in the reference was not differentially expressed. It is the first step in the production of endogenous hydrogen sulfide, H2S, and hydrogen sulfide is a really important signaling molecule. It's actually being studied as a way to put humans into kind of a hibernative state. It reduces the metabolic rate at low doses. At high doses, it's deadly. So it's kind of a balancing act there. And like I said before, two isoforms are differentially expressed between the seasons, but importantly, they're differentially expressed in opposite directions. So what do these isoforms look like compared to the reference? I'm just naming them as isoform A and isoform B, and then right here's the reference. So the first major difference we noted was that both of these isoforms retain the fifth intron of that gene. In fact, that fifth intron retention leads to a premature stop codon for both of those isoforms. The 13th exon is removed for both of those when compared to the reference. And then between the two, there is an alternate splice site in the third region, or in the third exon of this gene. So isoform isoform B has an alternative splice site. And that's really all we know about this gene at this point. All right, we know that it is cystathionine-Beta-Synthase when we blast it. But at this point, we don't know what any of these changes are doing to that gene. And so that brings me into my future directions pretty well, where we're going to hopefully functionally validate some of the isoforms we're identifying in primary cell culture. So pictured here are myocytes that were just cultured from grizzly bears this past summer. I got to go down to Berkeley and work with some researchers down there to bring that cell culture into the world, which was a lot of fun. And obviously, we're going to start with the genes with isoforms differentially expressed in opposite directions, because those are most likely the ones that have different functions from what is actually annotated. Now, I chose CBS, cystathionine, the gene, because it kind of highlights a problem that we have. And I'm hoping at a talk later today we can get into this. So this is how many isoforms were identified by PacBio. There were 35 total. And so actually going through each one of these and trying to identify which ones are biologically relevant is a massive headache. To do this for all 86 genes, it's just impossible. Right? So we really need new bioinformatic tools that are going to speed this process up and narrow down that list that we actually have to test. Like you can see this gene right here, willing to bet, or this isoform, willing to bet, not biologically relevant, but it still falls out in the PacBio analysis. And so I'm really excited by that. I think it's the 2 o'clock talk today where they're going to discuss these new bioinformatic tools. Really all I could think of in order to do this is proteomics. We're going to send we paired these samples, so we split each one of these samples in half, and we sent half of it off for proteomic analysis, so we can actually look and see, okay, are any of these isoforms being translated into protein? If so, then those are probably the ones that are biologically relevant. And with that, I'm going to go ahead and close. Does anybody have any questions?